I'm Pat Ryan with Carolina Biological. We're going to take a look at a lab today called Colony Transformation. This uses the pea green plasmid. That means it has a ring of DNA with a gene on it that causes bacteria to turn green. This green gene comes from a jellyfish. We call it naturally then the green gene plasmid. The materials that you're going to need to run this lab naturally include a set of directions. These are the student directions. 2 pages, front and back, where everything's well explained. They have questions for discussion and so on. You need 4 agar plates, Petri dishes, 2 of which have LB agar with ampicillin in the agar. The ampicillin acts as a poison to kill the bacteria. I find that using the word poison is much better than saying something like an antibiotic or a bactericide, because students sometimes lose those meanings, or like over their head, but they tend to understand what poison means. Poison kills things. Ampicillin is a poison that kills bacteria. The other 2 plates contain just what we call LB agar. LB stands for Luria broth. Actually, it stands for Luria bertolini. These are the names of people who made up these types of agar because they were the first ones that dealt with bacteria when they researched and studied them. And they found all the necessary amino acids, sugars, proteins, fats, carbohydrates, all the good things that bacteria needed to grow on. They have those things in the agar. It's a very good, rich, medium growth. You need 2 what I call the yellow loops. One's for picking up cells with, one's for picking up the plasmid with. And we'll talk more about those in just a moment. You'll need 4 of the transfer pipettes. The first one we'll use, we'll put calcium chloride into some tubes. Later on, we'll put some Luria broth. This is liquid Luria broth now, not the agar, into tubes. Then we're going to take some out of the tubes and put them on these plates we just saw. Some will go in just the LB plain plates, and then we'll use a different transfer pipette to put some solution onto the LB agar plates. You'll need 2 transformation tubes. These are very unique tubes. They have a double stop cap. You can see where the cap goes up and down, spins right around, won't come off. And then if you listen very, very closely, you can hear, when you push down all the way, you hear a You'll need a vial of calcium chloride, which needs to be kept on ice. So we'll put that baby right in there. And a vial of Luria broth. This again is the liquid Luria broth. Again, it has all the same things in the agar that bacteria need to grow on. It's a good, rich food for the agar. Keep that at room temperature right now. And one of the main ingredients, naturally, this tube with the green top, contains the green gene plasmid. There's a little bit of liquid down at the bottom. This contains 200 microliters. And there's just a small amount of liquid down at the bottom in which you find the green gene plasmid. That needs to be kept cold also. A type of rack to keep your tubes standing up in when you handle them. And then one of the most important items is your bacteria that you start with. Notice I have a Petri dish marked starter plate. One of the key things to make this lab really work good is to use cells that are 18 to 24 hours old. They're in what's called a mid-log growth phase. And they're much, much more apt to pick up the plasmids, take in the plasmids, and become transformed if they're in a sort of a young 18 to 24 hour growth period. I have a transfer pipette and a yellow loop I need to show you the demo. In each case you see that the instruments have a rounded end and a pointed end. With the transfer pipettes, we're going to open up the rounded end because the working end is the pointed end. They come sealed up, sterile, and the best way I've found, if you just get your fingers and thumbs up here, if just peel down until you're just in this kind of position with the heels of your hands together. And don't take the instrument out until you're really actually ready to use it. Again, in this case, we're using a demo situation here. There's your transfer pipette. Now these hold 1 mil of liquid up through the barrel up to this point, but we never use that amount. And notice that when I'm squeezing on this tube here, that's for getting the whole mill up there. But because we want small amounts, it's much better if you get your students to get used to practicing right here around where the barrel hits the end tube. Squeeze just very gently down this area. Even squeezing on the barrel is pretty good. And I would strongly advise the teachers to get a marker and mark the 2 places that you're going to need to use. Again, it's 1 mil, 1,000 microliters, all the way up to the top of the barrel. Halfway down is 500 microliters. In between, then is 750. And then quarter of the way down, or quarter of the way up now, I guess you say, this is the 250 microliter mark, and that's one of the settings that we do use for this lab. So it's always a good idea to have one transfer pipette marked that way, so you just take it around, put it right in your kid's face so they can see which line you're supposed to fill up to. Now the other place we're going to need does not have an individual marking on it, and I'm going to put little dots to make it different. It's right past where the real width of the barrel begins, and that is a 100 microliter marking. We use that at the very end to put the cells onto the plates. That's good to have, always set aside so you can show your kids the various markings there. The other instrument is the yellow loop again. Once again, a pointed end, a rounded end. This has a loop in it, and it's colored yellow, so hence its name. Again, open up the same way. Thumb and forefinger here, and if you just roll down till the heels of your hands are together, and again, do not take the instrument out until you're really ready to use it. And when you are, you take it out and use it. And the key thing is, anytime the students are finished using one of the instruments, it always goes back into the sterile container. Now that keeps you from getting all kinds of bacteria and different things on the table. And one final thing, because this lab has so many instruments and generates a lot of waste, you need something to keep your waste in. You can use an old beaker or whatever. But whenever the students are finished with the instrument, it goes back in the wrapper, simply put into your waste beaker, get it out of your way. Okay? So, let's get started. Using our lab marker, take one of the transformation tubes, put a plus sign on it. This will be the experimental tube. This is the tube that will receive the green gene plasmid. The other transformation tube, we put a minus on. This is a control tube that does not receive the green gene plasmid. One more thing I want to show you about these tubes, I'll use a demo tube here. One of the best things you can teach your students in biotech is what I call the little finger technique. We use so many little tiny tubes and vials and so on, and it takes a lot of manual dexterity to keep everything going. In this case, to use these, you wrap your little finger around the top. With the other hand, push up with your thumb and you sort of wiggle as you push up. Remember, these tubes are sterile just like the other instruments inside the wrappers. So this way you have the top inside your finger. It doesn't touch anything else and become contaminated. And your other hand is away from the lid so you don't get contaminants around the opening. The good old little finger technique. Okay? Now, one other thing before we get really started here. Notice the white seals on the caps. You're going to need some kind of a little blade usually. You can tear your fingernails up trying this. If you just get a little cut and get things started, and you peel it down, again, a lot of waste, that goes in the waste cup. Put that back on ice. And later we'll need the Lurie broth tube open also. The slice there. Peel down. Trash. Back at room temperature. Okay. First thing we need to do is add 200